In terms of inflammation, Silvestrol has been shown to inhibit the production of inflammatory cytokines such as interleukin-6 and tumor necrosis factor alpha. Necrosis is the death of body tissues. By inhibiting or interfering with the translation of their respective messenger RNA molecules, this inhibition of cytokine production helps to suppress the inflammatory response in the body. Overall, Silvestrol's mechanism of action involves targeting the translation machinery in the cells, leading to a reduction in the production of specific proteins, including those involved in inflammation. So, what is the mechanism of action of Silvestrol? Its mechanism of action involves targeting a cellular process known as protein synthesis, which is essential for cell growth and proliferation. Silvestrol specifically inhibits the activity of a cellular protein called eukaryotic translation initiation factor 4A, and this protein is a part of the translation initiation complex, which plays a crucial role in the initiation of protein synthesis. By targeting this factor, Silvestrol disrupts the assembly of the translation initiation complex and prevents the synthesis of specific proteins required for cancer cells survival and proliferation. So, the eukaryotic translation initiation. This factor is responsible for unwinding the structure region of messenger RNA molecules and allowing them to translate it into the proteins. Silvestrol binds to this factor and blocks its helicase activity, which is necessary for messenger RNA unwinding. As a result, the initiation complex cannot assemble properly and leading to a reduction in the translation of oncogenic proteins that promote cancer cell growth. Silvestrol's ability to selectively inhibit AIF4A makes it perfectly or particularly promising as an anti-cancer agent. Many cancer cells rely heavily on this factor-mediated translation to support their rapid growth and survival. So inhibiting this process can have a profound effect on tumor cells while sparing normal cells to some extent. This is the diagram of the eukaryotic translation initiation factor, in which Silvestrol EIF4A in the unwinding region and the translation initiation and also a codon is present. So what are the strategies to synthesize the Silvestrol? Although the total synthesis of Silvestrol is a challenging task, several strategies have been proposed to access this compound. Here I will outline two common studied approaches for the synthesis of Silvestrol. First one is the biomimetic totosynthesis and the fragment-based synthesis. So what is the biomimetic totosynthesis? This approach aims to mimic the biosynthetic pathway of Silvestrol found in the Algalia species. This strategy involves using relatively simple starting materials and applying enzymatic or chemical transmissions to assemble the desired compound. Researchers have identified the key intermediate rocagulate as a crucial building block in the synthesis of Silvestrol. By applying various chemical reactions such as aldol condensation, oxidative cyclization, and function group modifications, rocagulate can be transformed into Silvestrol. Although this approach shows promise, it still requires extensive optimization to achieve high yields and selectivity. Second one is the fragment-based synthesis. In this synthesis, it involves the stepwise assembly of small fragments to construct the target molecule. This strategy often relies on the identification of key structural motifs within the Silvestrol molecule and the development of efficient method for their synthesis. Researchers have identified the tetracyclic core structure of Silvestrol as a crucial fragment and have explored different routes to its synthesis. One such approach involves the construction of tetracyclic core through a cascade reaction involving cyclopropanation, ring opening, and oxidative cyclization. Subsequent elaboration of the core structure can then be achieved using various synthetic methods. So what are the structure activity relationship to its analog? So Silvestrol and its analogs belongs to a class of compounds which is called as rocacoalates and which selectively inhibit the activity of eukaryotic translation initiation factor 4A, an enzyme involved in messenger RNA translation initiation. And the inhibition of this translation initiation factor leads to the suppression of the protein synthesis and the subsequent indication of cell death in cancer cells. The structure-activity relationship studies of Silvestrol and its analog have focused on modifying specific regions of the molecules to improve its potency, selectivity, and pharmacokinetic properties. So these are the Silvestrol and its analog. Here are some key structural activity relationship findings. First is the airing modification, second is the beading modification, third one is the seeding modification, and the fourth one is the deading modification. So in the airing modification, the airing of Silvestrol is important for its binding to the RNA helicase AIF4A. Alterations in these regions have been explored to enhance its activity. For example, the substitution of the C2-methyl group and a C2-ethyl or a C2-phenylethyl group lead to the analogs with increased potency. So the beading modifications, the beading of Silvestrol is crucial for its binding to the AIF4A protein, and the modifications in these regions have been investigated to improve the binding of it and its selectivity. Introduction of various substitutions such as analogues and alkali groups at different positions of the bearings have resulted in its analog with improved activity. Seeding modification and the deading modification. The seeding of Silvestrol plays a role in stabilizing the binding interaction with AIF4A. Alterations in these regions have been explored to optimize the compound's potency. Modifications include the addition of substitutions or the replacement of seeding with alternative heterocycles. So the deading modifications of Silvestrol is involved in the formation of critical hydrogen bonds with the initiation translation initiation factor, and the modification in this region have been pursued to improve the compound's potency and selectivity. Alterations include the introduction of different substitutions or the replacement of the reading with the alternative moieties. So what is the stereochemistry? Stereochemistry is a branch of chemistry which is concerned with the three-dimensional arrangement of atoms and molecules and effects of this on the chemical reactions. So what is the stereochemistry of Silvestrol? So the stereochemistry of the Silvestrol and its analog has been investigated to understand its impact on the activity. Enantiomeric forms of the Silvestrol have been synthesized, and it was found that the stereochemistry can affect the potency and selectivity of the compounds. Overall, the structural activity relationship studies of Silvestrol and its analog have provided insights into the structural features necessary for potent AIF4A inhibition and anti-cancer activity. These studies have allowed the design and synthesis of analog with improved properties, potentially leading to the development of more effective anti-cancer therapies. There are the development efforts of the Silvestrol, which is first one is the anti-cancer potential, on the basis of the clinical and the preclinical studies. So it acts by inhibiting the activity of eukaryotic translation initiation factor 4A and RNA helicase enzymes involved in protein synthesis. By inhibiting this factor, Silvestrol disrupts the production of specific proteins which are required for cancer cell survival and proliferation. It has demonstrated activity against various cancer types including leukemia, breast cancer, and the lung cancer. Antiviral potential. In addition to its anticancer effect, Silvestrol has exhibited antiviral activity against a range of messenger RNA viruses, include the flaviviruses such as dengue and Zika viruses, coronaviruses, and Ebola viruses. Similarly to its anti-cancer mechanism, Silvestrol targets the AIF4A in viral infected cells by suppressing their viral replication. Synthetic analog development researchers have been working on the synthesis of Silvestrol analogs with improved drug-like properties such as increased potency, stability, and specificity. These analogs aim to enhance the therapeutic potential of Silvestrol while minimizing its potential toxicities. So what are the pharmacokinetic studies of Silvestrol? Silvestrol has presented challenges in terms of its bioavailability and toxicity. Researchers have been investigating strategies to improve the pharmacokinetic properties of silvestrols, like enhancing their solubility, stability, and delivery to target tissues. In vivo studies, silvestrol and its analogs have been evaluated in animal models to assess the efficacy, safety, and pharmacokinetics. These studies provide important data for the potential translation of silvestrol into clinical trials. So this is the latest research on the silvestrol and its analogs, and this is the graphical diagram. With the help of these diagrams, we can be able to see that how much the researches are going on the silvestrol and on its analogs. And this is the link of our article where we can be able to see the whole topic and overview the silvestrols. So in 2016, an CRISPR mediated drug target validation reveals selective pharmacological inhibition of the RNA helicase. AIF4A inhibitor Silvestrol blocks the growth of human glioblastoma cells by inhibiting AKT, mTOR, and ERK1/2 signaling pathways. In 2022, in silico biochemical and in vitro analysis of silvestrol binding to the dead box RNA helicase AIF4A reveals broad antipathogen potential of rocagulids across the eukaryotic tree of life. In 2022, and comparison of broad-spectrum antiviral activities of the synthetic rocagulade CR31B and the EIF4A inhibitor silvestrol in 2020. Broad-spectrum antiviral activity of AIF4A inhibitor silvestrol against corona and the picornaviruses in 2018. This is the end of my presentation. Thank you so much for your interest and attention, and please do subscribe and share the video.